All right. Hello, everybody. It's super good to be here today to talk about this topic.
Well, hello, everybody. So my name is Stephen Harlan, as lovely introduced. Yes, I work with protein interactions within photoreceptors, and I recently picked up Blender about 2 years ago and sort of self-taught myself, just sort of playing around with it, trying to visualize different things in 3 dimensions. And through that, I was able to hone in my skills and become very proficient in it, which I'm very excited to talk to you all about today.
And my name is Helena. I work in the Sternodka lab researching antibiotic-resistant proteins using cryo-EM and crystallography data. So I've had a lot of experience visualizing this data and trying to create effective figures for my work. So we're going to explore why science visuals matter, how to design them effectively, and the best tools for this job. So as scientists, we focus on experiments, data collection, and analysis, but communicating our findings can be just as important because data only has impact if it's understood. Now, as data grows more complex, visualization can be a key to interpretation. With increasing data volumes and more accessible visualization tools, the challenge isn't just analyzing your data, but how to present it. And so this highlights the need to kind of educate on how to read, interpret, and design your data responsibly, because clear, well-structured visuals can make the sense of the science that shapes our world. So this is what our presentation will be covering today. In this first part, like they mentioned, we're going to go over principles of science communication, some rules for figure design, and then I'll go through my favorite tips and tricks for Illustrator and Chimera X. And then after our Q&A, we'll go through using Blender. And Steve will have an awesome kind of walkthrough for you. And by the end, we hope that everyone has kind of a solid toolkit for creating clear, impactful science visuals. So to first take a step back, why should we care about communicating effectively with visuals? Looking at the big picture, I think the COVID-19 pandemic was an awesome example of just how important science communication can be. Navigating public health policies and understanding biotechnology, like the mRNA vaccine, would have been really difficult without effective visuals. And a standout example of this was the concept of flattening the curve, a simple but powerful visualization that helped the public grasp why behavioral changes were needed. And this directly influenced kind of public health. So this highlights the fact that well-designed figures and schematics aren't just for experts. They make complex topics understandable to a broad audience, whether it's the public or policymakers. Science visuals can kind of bridge this gap of understanding. And graphs and figures are one of the main ways we communicate our science. They share how we they shape how we interpret information, influencing opinions. However, it's easy with visualization tools to kind of muddle up the data. And here I have an example of a group that created a graph to argue a specific kind of narrative about a group in the US. And at first glance, this looks convincing that these two things are kind of inversely correlated. But when you look at it in the context of a greater data, you can see how this was a bit of a visual manipulation and is distorting the actual story that the data tells. And this kind of just highlights our responsibility that as scientists and data communicators, we have to prioritize clarity and accuracy, because poorly designed or misleading graph can misinterpret the results. So our job isn't just to visualize data, but to communicate it honestly. And so now I'll just go through a few examples of what are some good graphs to communicate certain types of data that you might have. Now, bar charts are great for numerical values of sets that should start around 0. And this is in contrast to dot plots, which are good when you're looking at small differences between data sets. And how you organize your data in this figure also matters in terms of here, when we organize it according to ascending, we can immediately see some trends in the data that the kind of example, the red example on the left doesn't show. Color can also be a really powerful tool. And here it can be used in a heat map, which is how we can communicate numerical values for sets of categories in different groups. However, here we see one rainbow and one more two-tone scale, and more on this later, but rainbows are not always the best way of communicating a progression of data. So this kind of highlights that example. Now, looking at different distributions of data, histograms can be a great way of showing trends, but I would here, this kind of points out that the binning you use on your histograms can be really important for making your data kind of clear and easy to understand. Box and violin plots are fantastic for showing the summary statistics, such as the mean and the range. And violin plots can show the kind of shape of these distributions. And science journals are often moving away from showing bar plots and towards showing box and scatter plots. And ridgeline plots compare several distributions at the same time over a horizontal axis without overwhelming the image, which can be great for showing distributions over time. Now, moving on to proportions. Pie charts are mainly good for very small data sets because they can show differences in the proportions when the sample sizes are small. Side-by-side bars are very often used in science, and they're great for showing numerical values of sets of categories when there's kind of trends you want to highlight. And then stack bars and densities are often shown, but when you start having more than a couple of categories in these, they become very hard to follow. But they're great for when the sum of the amounts is important. And when we start having two or more variables, scatter plots show the quantitative relationship between two variables. And when we add another factor in there, we can show heat maps that add color into this to show patterns and associations. And slope graphs can show a two-step change of variables, which can be used for continuous or categorical data.
Now, just to hop in and touch off of what Helena just described, if you look on the left, here we have a 2D representation of a photoreceptor, which is something that I work with. So I'm very passionate about this topic, and I can talk all day about it. And while this provides the basic overview of the structure, it does lack the depth and sort of spatial context to make it difficult to fully understand the relationships between these different cellular components, as it looks like quite a strange structure. And I often found that these representations are inadequate when considering my research, because biological systems are inherently three-dimensional, and without the ability to visualize their spatial organization, it's challenging to truly grasp how complicated these systems can be. So if we take a look at this 3D model, we can really appreciate the complexity of a system such as this, as the intricate stacking of these disks do arrange in membrane structures and their overall organization become much clearer. Now, this sort of different perspective can not only improve our understanding of their function, but also provides a more intuitive way to conceptualize these outer segment architectures in photoreceptors. So this is just one unique example where 3D models can allow us to communicate these concepts more effectively, whether it be for publications, presentations, or like educational materials, for instance. They offer this dynamic way and interesting way to not even just explore data, but also explore the reasoning behind the data or help explain it.
And in science, we often rely on 2D graphs to explain or convey complex relationships. Like often in kinetics, we see this with Michaelis-Menten plots. And here, for example, we have a plot of temperature versus kcat that I'm going to show in a second. And while effective, static visuals can sometimes fall short in capturing the full depth of a biological process. By introducing another dimension into this, by having a 3D dynamic model and a video of this enzyme's active site, we can add a new layer to our understanding. And so here we see that as temperature increases, molecular motion increases, highlighting how structural flexibility plays a role in enzyme function. Now, this visualization kind of gives us another layer of how and transforms what would otherwise be just a trend to memorize to kind of understand something more tangibly and intuitively. And this illustrates a broad principle. Good visuals are not just supplementary, but they're really essential in trying to explain these very complex interactions, cellular structures, structural data, and well-designed graphics, especially 3D models and animations, can kind of make this complex data accessible. So that's kind of one of our main points we wanted to make, is that science visualization is not just about aesthetics, but it's a tool that can help expand your work's impact. When you have visually appealing proposals, they can help secure funding for grants. You can potentially open up collaborations across disciplines if your figures and data are easy to understand. And especially when you're coming up against a word limit in a proposal or something like that, putting in a figure can save a ton of word space and allow you to communicate a concept that otherwise you couldn't fit in there. So with all of this being said, I'm now going to start talking about what are some guidelines for making good science visuals. And we'll start in 2D, work our way up towards more complex data, and end in Blender. So in figure design, how do you draw focus? How are you thinking about what is the main emphasis? Now, one good way to do this is with color. Keep most of your things a general color palette, and then you can have a contrasting or highlight color. That's a great way to draw people's attention. Additionally, size in a figure can be a way to draw attention and highlight a certain aspect or key concept that you're trying to communicate. Clarity is really important. Again, also to kind of simplify your figures as much as you can, so they're only communicating the most important things. So here, when we compare A and B, A is kind of unnecessarily complicated with a lot of arrows, versus in B, we can kind of intuitively see what's happening. And so when you're creating your figures, ask yourself, what are the essential elements? Is there anything missing? But what is unnecessary repetition or decoration? And then on the right, we can see another example. And here, maybe a more kind of visual figure. You want to make sure that everything is labeled the first time you see it, especially, and that there's enough structure to keep it organized, but not to have it too cluttered. The next thing to think about is accessibility. Now, there's a couple of pretty basic rules you can keep to that create or make your figures accessible to people with color blindness or accessibility issues. So make sure you have good contrast in your figures, using black text instead of colored. Try not to have colored boxes behind your texts, and avoid red and green color combinations. And then you can use a tool to check for color blindness and contrast as kind of just a step in your workflow to make sure that your data can be seen by everyone. And something that I also mentioned earlier is the importance of color scales. Now, in science, rainbow is kind of a standard color scale to use when communicating a range of data. But I'm here to tell you that it's not usually the best way to do it, because in a rainbow, there's these kind of two peaks of intensity of light colors that our eyes tend to be drawn to. But those aren't a true, so it kind of isn't a true linear scale. But there's scientifically created color palettes that make up for this and have a correlation between the intensity or lightness and where you are on a color scale. And just to really prove the importance of this, if we take a grayscale image here, or example, and we put a rainbow color scale on it, it becomes almost unrecognizable for the image of the face and the image of the of Earth behind it. But when we put it on one of these scientific color scales, it's just as easy to interpret as the original grayscale. So now when you're thinking about doing this with data that obviously doesn't have an original reference, make sure that you're considering how your color scale is interpretable. So those were kind of the basics of picking some graphs and charts and some design rules for your figures. But now we're going to get into the meat and potatoes of using these programs for making figures. And I'm going to spend a lot of my time talking about Adobe Illustrator. Now, Illustrator is kind of the professional standard of, I think, making science figures, and most journals often ask for your figures to be in that format. There are, unfortunately, it is a paid program, and I'm going to go over a couple of free alternatives, but because most people that we know in our field use Illustrator, that's where I'll spend most of my time. So in order to understand Illustrator, we're going to have to talk a little bit about vector art. Now, most photos we see are made up of pixels, and that's called a raster image. Vector art instead is made up of vertices that contain information about shapes, lines, and colors in relation to each other and not in relations to pixels. And why is this so important? Because then when you scale it, pixels get pixelated, but vector art is scalable and movable and editable in a way that doesn't change its quality. It also lets you layer art on top of each other and change transparency. So it's a very powerful tool. Now, to kind of look at an example here, this is from the Illustrator tutorials. We have a crab, and it's made up of different shapes. And each one of these vertices is movable and editable. Now, we can kind of change its size, change the angles, the roundness. And so when you're making your figures out of vectors and shapes, you have to think about the concept you want to communicate and how to break it down into individual shapes that you can then edit. And so obviously this is an abstract picture of an animal, but when we look at something maybe a bit more scientific, this is a figure that I made that is also made up of vectors in this program. So each phospholipid is a little circle with some little squiggles that's transparent, and you can put in structures and create this very kind of clean looking figure. Now, there's hundreds of hours of tutorials about Illustrator. It's a professional program made for graphic designers, so we won't have time for a full tutorial. However, I will say that I find it very worth learning Illustrator through just trying to follow some of these graphic design tutorials, try to make little artwork that you can think of, and over time, it'll become a program you are comfortable in. But I will focus on my kind of favorite tips and things that smooth my workflow and make my time in Illustrator efficient, and that are related to scientific figure making. So to start talking about Illustrator, we're going to go into just a little crash course to orient you, so some of these things make a little bit more sense. Now, when we're in Illustrator, we have a select tool, and your tools are on the left. You have some properties windows on the right, and the tabs on top of which figure you're in. So the selection tool, when you click on it, you can then click on an object and move it around. You can also scale it like you would in a PowerPoint or something you're used to. But the first thing is that when you scale in Illustrator, it doesn't preserve your ratios, which means it'll get all drawn out, which isn't great. So you have to make sure you click Shift when you drag in order to preserve your ratios, which is maybe the most useful shortcut you can have for Illustrator. In order to keep your figures organized, I'd also really recommend using layers. So here, the protein's on one layer, and I'm going to make a layer above it that I'm going to put my labels on. And this lets me lock the protein layer so it doesn't move around and I can't edit it. And I can just focus on my labels, which can be great for keeping your artwork organized. The type tool lets you put in text. And then once you add text into your image, it's scalable and editable in the same way as any other object. So here I'm going to shift drag again. And now in this example, I'm going to try to add a little arrow. Now, arrows are kind of hard when you're starting Illustrator because there's a lot of ways to add them. There's a pen tool, a curve tool, and a line tool. And they all do similar things, but slightly differently. So here I pick a curve tool, and it's going to add a curve that's going to have some properties. It's going to have a fill color as if it was kind of the start of a shape, and it's going to have a stroke width, which tells you how thick the line is. So I'm going to take away the fill because I don't want this to be a shape. I want it to be a line. And then I'm going to increase the stroke a little bit so it looks like a line. And now when you double click on the stroke name, you're going to be able to add an arrowhead. And there you go. That's how you make arrows, and you can edit where they go, change the colors, scale them. And that just is maybe not completely intuitive. If I want to add a line here, a straight line, I can also use the line tool. And then here again, I double click on stroke and I add an arrowhead. So that's kind of a super basic figure labeling. And here I then just show that in the kind of home of Illustrator, there's lots of tutorials that you can watch and go over. And additionally, on YouTube, again, there's lots of resources to get started in this kind of complicated program. So moving on, I don't know if that. Okay. The backbone of your organizational system in Illustrator are artboards. And you can think of these kind of like pieces of paper on a desk where you're putting all your figures. And here I have the figures laid out for a paper. And on the top, I have my main text. In the middle, I have my extended data. And on the bottom, I have my supplementary. So this just kind of shows you that you can have your figures organized in this, your kind of desk space in a file. And you can have them all in the same area, which can be quite helpful. Now, when you open up Illustrator, the first view you get is asking if you want to make an artboard and what size you want it to be. Now, the artboard tool lets you change the size of your kind of piece of paper that your figure is. If you double click on it, you can change the width and the height. Now, something that's super helpful is to look up the specifications for whatever journal you're thinking of submitting to and change all your artboards to that size right at the beginning, because that way you'll have that organized and ready to go. Now here I'm showing another fun part of artboards, which is that when you have some objects on an artboard, you can copy and paste that artboard itself, and it will create a duplicate that has all the same labeling in the same spots. Now, if you're making kind of iterations of figures, this can be great because you create a copy with everything in the same place, and then you change one thing to look at how it looks, or you can make a series of different figures. And I find this quite nice. So here I'm just copying this artboard with Command C, and then I'm pasting two more right next to it, and it kind of preserves the annotations and the formatting on them. So now I'm putting my other two, or making my 3 series of a top, side, and a bottom view, and they're nicely organized, the correct size, with all of the text in the same spot. And like I said, I really can't emphasize enough how nice it is to to kind of go through your figure requirements beforehand, and then use that going forward. A reason for this is that often there's a minimum or a maximum text size. And when you're editing in a kind of randomly sized artboard that isn't, you'll have the text on there and you'll try to make it the right size, but you don't know if you're in the right specifications. But if you set your size to the correct physical size, then when you're editing your text, it's the proper proportion to the figure. And that should save you some time. Another great little part of artboards is that when you have them in the same document, you can export them really cleanly and rename them. And so here I have the artboard named top, and then I'm going to go and I'm going to name the other ones side and bottom. And when you click on it in properties on the right, you can do that. And then when you go to export them, you can go to export as, and you'll export them as individual PNGs using the artboards. So you can see that on the bottom here. I'm going to click the use artboards box. And then when I go, you can actually specify a range if you want only a couple or you want all of them. I clicked all. And then when I go through and I export all of them, they're going to show up in a folder labeled according to the name of the artboard and the file that they came from, which once you start having a lot of figures, can again help you kind of keep everything organized. Artboards also let you put a grid on, which can be great when you're going through and making figures that have a lot of things to be aligned, or when you're going through your A, B, C, D paneling. A great thing is to turn on this feature so you can make sure all your things are properly aligned. Now, we've already talked about colors, so I will now talk about them in the context of Illustrator. A good color scheme is great for having engaging and cohesive presentation, or for keeping your kind of figures looking cohesive. So there's a couple of resources that I like using. A software called Colors can, with 2 O's, online, can let you go through lots of different options, and you can go through and like one by one pick random, have like a palette generator, which can be nice. And Illustrator also has a built-in color guide that helps you pick a palette depending on a certain color. So here, for example, I picked a pink color, and then you go to a submenu of window and color guide, and then you get into this window and it gives you a bunch of options for different color palettes based on harmony rules. So, and this also works if you select more than one color, and it creates kind of complementary or split or like monochrome. So you have lots of options. And Illustrator also has a built-in color palette library if you need inspiration. Now, color schemes in Illustrator can be organized through a really useful tool called color swatch libraries. Now here, I want to make a 6-color palette. And I'm actually just showing a fun trick I like to do where I use the eyedropper tool and I select a color from maybe a different figure I like or a gradient I like. This is a really fast way of kind of importing colors into Illustrator. You can just screenshot something from your computer, put it in Illustrator, and eyedropper them. Now I select all of the objects that have the colors I want, and I go to this color swatch library. I click on new color group, and then I put include the swatches. And just like that, that color library is imported and I can use it to change the color of something else. So here I want to change the background. I double click on it. And there in the bottom is my color library ready to go, which can be a great time saver. Now, another kind of fun feature of Illustrator is that you can recolor an entire artwork to be a certain color scheme. So here I have a figure of a mechanism, and I go to this recolor window, which opens, and then I click this color theme picker, and I can click on a certain object, and it'll recolor everything in that figure to be in the same tones as a certain color palette, which is pretty nice. You can also unlink these individual colors and change them one by one if there's a certain one on there that you don't like. And if you're trying to, keep your figures cohesive, or you change your color library at some point, this is a great way of really quickly editing colors and making figures cohesive. Another cool, powerful tool that Illustrator has is called image trace. And in image trace, you can take an image for here. For example, I'm grabbing a figure on antibiotic resistance, something I work with, and I'm going to make it editable. Obviously, when you're doing this, you want to cite the original figure properly because you're using it. So it's not just your artwork. You need to give proper citation, but we can edit it and change the colors. So here I'm going to image trace, and I'm clicking that I want it to be traced in 3 different colors. And this creates this artwork in vector format in the amount of colors I tell it. Here I lost some of the kind of information I wanted, so then I went to 16 colors, and that preserves a lot of that original detail I wanted. However, I want it to match my colors that I have already picked. So after, I'm just going to go to recolor the artwork as I just showed you, going to color theme, and just like that, I can have it in my color palette. Now, because this has been turned into a vector image, that means it's composed of shapes, and shapes are editable. So I can expand it into all of the shapes that it's made up of, and I can delete the shapes that make up the background. So like this, I can delete backgrounds very easily, no tracing necessary. It kind of does it for you. And now you can put it over slides or backgrounds that are not white, and it can look really clean. Now, starting to talk about structural files. I'll get into this a little bit more later. This works with any structural files, not just Chimera X, but I'll just go with Chimera X initially. Now you can save your structural files with a transparent background, usually in the command line. So I'll show this here. I'm going to save a certain view for a figure I want to make with a file path, a name, and then this information that tells it to have a transparent background. And I would urge everyone when they do this to save a corresponding file of the view in Chimera. This is really great because then if you ever want to edit that exact picture, you'll have that file already saved in Chimera X. Now I'm going to drag into Illustrator this image from wherever I saved it, and I'll have a great transparent background image that I'll show you something cool you can do with later. Now, when you're going to go and annotate an image, sometimes you need to put text over parts of your image because maybe there isn't room for you to write it next to it. Now, my favorite little tip for doing this is to use a Gaussian blurred white oval. So you put an oval underneath your text, and you use this special blur effect that diffuses out that color. And what that ends up looking like is that your text is readable, but the information under it is still readable as well. So this can be great for labeling. Now here I have a fully labeled structure that I showed earlier in Illustrator. But as very often happens, sometimes you make all your figures and then you go and you discuss it, and you decide, or your PI decides, that one thing has to be a slightly different color. And so this can be often a pretty big time hassle to change, but not with linked files. So I'm going to go back to that file I had opened, this view for this figure. And now I'm going to change the color of this kind of primary residue in it, or my focus of it, to be purple. I'm going to change it so I can see the atoms in the right colors. And now, in a kind of key move, I'm going to resave this over the original file path. You do lose that original file path, but usually if it's just changing one color, it's pretty easy to get back. So I'm going to save over the file path, and then in the same thing, I'm going to save it again as a separate Chimera file. And now when you go back to Illustrator, it will recognize that the file is different, and it will auto update it in Illustrator. And so all of your labeling, all of the views is in the same spot. It will just be the right color. This does come with a small risk that when you change where your file is located, that really confuses Illustrator because you break that file path. So when you use this kind of tip, you have to make sure that your files are very organized and you're not planning on moving them anywhere, because when you move them, it'll break this file link. Additionally, before you submit it anywhere, you have to embed those files, because they are Illustrator's getting that information from that file path and not saving it in Illustrator. So somebody on a different computer can't open that file. And there's a way to do this. You just go to window links and then you embed the files, but it just be aware that this is something you have to do if you're just dragging and dropping files into Illustrator. Very small tip that I found useful is that if you're working with chemical structures, the easiest way to get them into Illustrator is simply to copy and paste them from ChemDraw into Illustrator, and it saves all of the vector information. It saves the text as editable text, and I just like that little tip and wanted to share it. And so looking at all of these tips together, I thought I'd put like a little example as to how efficient your workflow can be. Here I have a schematic I want to show, and I'm putting in an image of an X-ray synchrotron in order to finish this figure. Now it has a white background that I want to remove. So I'm going to image trace it into 16 colors. That's usually a good one to pick. And I'm going to delete the background off of it. And that will give me a no background image. And then additionally, looking at this picture of the synchrotron, it doesn't really match the colors that I already have on there. And so after I scale it, well, I'm going to recolor it with the colors from the black and gray in the other parts of this figure. So I'm going to color theme picker, click on that, and it's going to make it the appropriate colors that I want to just graphically look nice in this example. And in this X-ray synchrotron X-ray data example, I also have a mesh view shown here that is kind of the wrong color to match the other electron densities. So I have loaded up a view of that I had saved, and I want it to match the color better. So I'm going to color the atoms in this view to be that different color, a darker color, to match my figure better, and show kind of the representation that I want. Then I'm going to do the same thing I showed you, and I'm going to save over the figure. When you click the up arrow in Chimera X in the command line, it'll pull up your most recent commands, which is also great for kind of saving some time. And it has already auto updated them in Illustrator. And just like that, I'm able to kind of quite quickly get the kind of cohesive look that I want. So I have spent a lot of time talking about Illustrator. Thank you for bearing with me. I'm going to go over 2 free alternatives now. So BioRender is a company that makes pre-made science vector art, which is great. They're a bunch of science illustrators and have a huge library of different pre-made scientific options. So it's scalable, their free trials decent, and they have lots of science tools and graphics that you can, and templates that you can use for your research. I primarily like using BioRender for my workflow, but I also use it to get inspired and see how their science illustrators made different vector art. The cons is that you have to work in their workspace, which is sometimes, well, it is not as powerful as a tool like Illustrator. It requires a paid subscription if you want to use their premium designs, and it's also not quite as customizable or editable, but a great option when you're starting, and I still use it for my some of the equipment. Another option that is an Illustrator, is comparable to Illustrator, is something called Inkscape. It's just a free vector-based software, and you can directly import Adobe Illustrator files or vector files as well, which is you can submit straight to journals. So great if you don't have access to an Illustrator subscription. So we've talked a lot about good figures, and I've talked a lot about Illustrator, but one of the kind of reasons we're here is to talk about structural data, which are complex molecules at an atomic level. So how do we visualize them? So here we have the example of a surface model. We can have a cartoon model, and we can have a stick model. And what this data really is are vertices and atom locations within 3D space. And so the 3D modeling software that we're going to go over now are ways that you can visualize this atomic data. Now, PyMol is a great general program that's been traditionally used, and it has lots of scripting you can do in it. But the free version is limited and the scripting does take a bit of a learning curve. Qt is wonderful for model building and refinement, but it does have limited rendering and an older interface. Chimera is the what Chimera X was originally based on. It's good for molecular analysis and has some tools that Chimera X lacks, but it's an older interface and it's no longer being developed because the focus is on its successor, Chimera X, which was made specifically for cryo-EM and high quality visuals. And it's supposed to be kind of intuitive for cryo-EM data. I will, as somebody who works with cryo-EM data, go over just a couple of useful things that I find in Chimera X. Chimera X has a command line, which can be great for coloring structures. And can kind of really speed up when you need to put information instead of using the graphic interface. So here I'm showing how you can use the command line to either color different selections or to specify residues that it colors. And when you open up a structure, oftentimes you want to look at it in the same coloring. If there's a certain residue you want to visualize, it's often the same when you're repeatedly looking at a structure or opening up in different views. So you can combine all of the lines of commands to color a structure a certain way in a text editor, just regular text edit on your computer. You save it as a .cxc file, and then you can just drag and drop it into Chimera X, and it's going to run through all of those commands and color your protein exactly how you want it. And so when you're going through and refining your data or iteratively going through different let's say chains or slight mutants or something like that, this can really speed up your initial view when you open Chimera. Additionally, this might be kind of a pretty well-known feature, just to make sure that everyone knows that you can color densities, your electron density in Chimera, as well by the atoms around it, and which is a great way of kind of transferring that color information you had from your PDB into electron density. Chimera X can also do electron density maps as mesh, which shows the kind of quality of density around residues, which is often necessary in some parts of your figures. And there's a nifty command called subdividing your mesh or your volumes, which will give you kind of a prettier looking mesh, which can be nice for these figures. And lastly, there's if you do want to visualize crystallography data in Chimera X, you can convert .mtz maps into density maps with a program called CCP4 FFT. So know that that's an option for you. And there's also a package called Clipper, which can be installed and allow you to visualize .mtz files. So with all of these kind of programs for visualizing science data, these are kind of like by scientists for scientists. There's now the additional program of Blender that will be the focus of next section, which is an advanced 3D rendering and animation software that gives you a ton more control, but just is kind of harder to get used to. So Steve will give us a wonderful tutorial on how to visualize your structural data in Blender. And with that, we'd be really happy to take the first round of questions.
Thank you so much, Helena. So if you have a question, you can type it into the Q&A that's at the bottom of the page, or you can raise your hand. Someone asked, can you use Chimera X for non-Creo EM data?
Is my audio still on? Yeah. Yes, you can use Chimera X for any sort of density data. Like I was saying, the .mtz files are what crystallography data comes in. So you can use it for that, and they can generally, it's compatible with most types of structural data.
And we have one person who would like to ask a question. You can go ahead.
Yeah, okay. Thank you, Helena. I want to understand how to really like translate heat maps. I see them all around, but I'm not able to really understand the data they are actually telling. Thank you.
Yeah, heat maps can kind of show, I think, a lot of dimensions of data. So often when you're looking at the relationship between more than two variables, heat maps can allow you to see patterns. But the main thing for heat maps is picking a good color scale, because if you're looking at kind of the you have to choose if your data is like from just one side to another, or if it's kind of multimodal. There's different resources you can look at to see, depending on your data, what color scale you use to show it. And I think that's the kind of most important part of interpreting a heat map.
Thank you.
And we have another question in the chat. What about VMD for 3D structure representation?
Does that mean like molecular dynamics?
Yes.
Oh.
Yeah, or yeah,
Yeah, so I feel like I can help answer that. You can actually use Blender for that. I unfortunately do not go into that in this presentation, but as I mentioned when I start talking, the person who created an add-on for Blender to work with molecular data, his name is Brady Johnston, and he has a wonderful YouTube tutorials all on this topic, and he makes it very easy to understand because I don't work with molecular data too much, or sorry, molecular dynamics data, but yes, it's actually quite simple in Blender.
And yes, I also don't work with molecular dynamics. It's my understanding that Chimera X is capable of a lot of these 3D simulations. And it's all free too, so you can try it out. But I do think the more advanced molecular dynamics simulations are often in paid software.
Perfect. Thank you. And another question was, do you have recommendations or tips for numerical data beyond Excel?
Yeah, I do. So to maybe slightly go back here. Excel is obviously a good place to start. RStudio is also a free and really powerful tool specifically for kind of science data. It does involve having to learn a little bit of coding or just really learning the language that R uses. There's tons of really good tutorials for R because it's very commonly taught in university for science or for biology. So R is great for kind of like microbiome data or just kind of biological data visualization. MATLAB is more physics-based, I would say, or kind of engineering focused. And then Prism, if you can get a license to do it, you do have a 30-day free trial when you go use it, is amazing for making, working with data like mouse data or working with, it's kind of like Excel, but made for kind of experimentalists or biologists, and it makes a lot of the figures you see in papers are often made with Prism.
Perfect. Thank you. And someone else, Carol asks, how does your process of planning figures look like as you collect data and or start writing a manuscript?
That's a great question. The first thing that we did is you kind of map out, often just on paper, you draw out some initial figures you have in mind. The main thing with a figure is you have to know what you're trying to say in it. So if you're looking at, you want to do an overview of your structure, what do you want to highlight? Do you want to highlight the electrostatics? Do you want to highlight the a ribbon model or a surface model to show, you know, the convexities or something like that in it, or a binding site? So I would say really try to figure out what is a piece of information that you want your figure to share. And then from there, you can, it's a structural model, go through and try a bunch of different views to see which of those is communicating that concept the best. And don't get attached to your first draft of your figure because there's like 100% chance it will be different in the final draft. So be okay with criticism or with kind of constructive feedback because you'll probably need to change it as you're working through creating a visual that's easy to understand. I couldn't tell you how many drafts I had of that kind of initial pathway figure, probably at least 50. So yeah, just to get, it's iterative and get lots of feedback.
Okay, thank you. And one last question before we have the next presentation. When loading crystallography data in Chimera X, is the map threshold in the volume viewer the same as the sigma that you would set when working with the MTZ file in PyMol?
I'm actually not sure about that. I think I think it is, but you might have to double check that. It should be though.
Okay, perfect. Thank you. If anybody has any more questions, you can type them into the Q&A box below. And as the next presenter presents, maybe Helena can answer some of those questions.
All right, am I good to go? Yeah, okay. Well, I am very excited today to talk about something that has been quite a passion of mine for the last little while, and that is Blender. Now, Blender is a free 3D rendering program that allows you to make these photorealistic advanced materials. And in terms of the biochemical context, sometimes you want more control over what you visualize. So unlike traditional molecular visualization tools, Blender allows us to create these photorealistic materials, making models not only scientifically accurate, but also visually stunning. So with Blender, we can also produce these cinematic animations that provide this dynamic way to communicate these complex biological processes with both clarity and impact. Now, one of Blender's biggest advantages is its ability to generate these repetitive molecular structures, such as DNA helices, lipid bilayers, and protein assemblies, using what they call procedural modeling. Now, what this is, it's a non-destructive approach that makes it easy to refine and change different aspects of your model without actually having to start from scratch. It can also handle large data sets more efficiently than these traditional visualization tools, allowing us to work with this high-resolution molecular model while maintaining interaction in a 3D space like you can do with PyMol or Chimera. So to look at some examples, here we can see this in action where this sort of protein complex can come together, illustrating how molecular interactions drive biological function. We could have fun cartoons, like something being plugged in. We could have a DNA polymerase unwinding a segment of DNA. We can showcase self-assembly mechanisms as protein dimers assemble around a disk. And you can even draw up some methods such as cryogrid staining or even picking up glass cover slips. We can visualize a ligand docking into a protein's binding site, an enzymatic reaction. And even for fun, we can look at a base editor of CRISPR-Cas9 with guide RNA being highlighted and base being edited there. So this is just a quick look at just some of the animations you can do and how visually appealing they can be to kind of drive forward this notion of understanding and communication. Now to dive right into it, how exactly does Blender work? So to first provide you just the very basics, when you first open up Blender, you are greeted with this 3D workspace that includes a camera, a light source, and a default object. This could be anything, however, it is usually a cube, for instance. And this setup is the foundation for building and rendering these 3D models. And to look at objects a little more in depth, these contain specific vertices, each with coordinates within a 3D space, each having an X, Y, and Z coordinate. And a mesh is able to connect these multiple vertices to form the surface of it or a face, for instance. So you can see if we manipulate these vertices, they can actually change their coordinates. So that's sort of the basis. And now how do we exactly handle molecular data in Blender? Well, to help answer that, and as I briefly mentioned in one of the questions, perhaps one of the biggest advancements in the visualization in Blender for biochemists has been this development called molecular nodes, which is an incredibly powerful tool developed by this man, Brady Johnston. And it allows us to easily upload and manipulate PDB files and even AlphaFold files directly within Blender. So as you can see on the right, that's kind of the basic principle behind it. It uses Blender's built-in geometry node system, which essentially serves as there's an input and an output, and you can connect styling options in between there, such as a cartoon representation. And you can also select specific properties of it to be shown that way, and you can customize different materials. So this gives a researcher full control over how their structures are visualized. And using this, we can move beyond traditional visualization tools and take advantage of Blender's advanced rendering capabilities. But how exactly does it work with molecular data? So similar to the object we looked at before, every atom in a protein does have this precise X, Y, and Z coordinate that is displayed in the PDB file or AlphaFold. So here, if we start with a single amino acid residue with roughly 5 atoms, we can get its atomic coordinates displayed, both its X, Y, and Z, and we can import this data into Blender and reconstruct the protein atom by atom, placing each in its spatial position. So as we kind of start to look at more of the atoms within it, we see the structure start to form that may look a little familiar. Now that protein is actually green fluorescent protein, or GFP, which I'm sure a bunch of us are familiar with. And this was done using the PDB file 2AWK, this image right here. Now this may look a little daunting as to how do you even begin to make this, but Blender makes this very simple, and we can break this down into 3 different styles. So we have a chromophore, a backbone, and the electron density, each styled in their own way. The chromophore being a ball and stick, the backbone a cartoon style, and the electron density being a surface. Now, before we actually can play around with it, we first have to upload a PDB file into Blender. And what does that look like? So molecular nodes makes this very simple, as you just simply type in a PDB that you're interested in. You can choose the style that it gets imported in as, and you can center the structure, hit fetch, and boom. You can then scale it with S, move the cursor, or rotate it by using R, just hitting R or S on the keyboard. So that's a very simple way to import it. Once it's imported, how exactly do we go beyond that and start to play around with how it looks? And that is where molecular nodes comes into play. So if we simply divide the screen into 2 and hit geometry node editor, which is the software that molecular nodes uses, we generate this workflow here. So the group input represents the raw molecular data from a PDB file, each atom's X, Y, and Z coordinates. And we also get additional properties like element type or bond connections. And the output defines what we see in the viewport, and we can then style it however way we want to by interrupting this connection. So now if we switch into an edit mode, we can see this process take place where each atom's vertices that is in 3D space actually get styled as a sphere around it. So it just takes all those, puts a sphere around it, and that is the fundamental building blocks of a 3D model here. And if we look at this, there's only one node for the output. There's only one connection. So in this example, we can either look at either these atoms as just their vertices, or we can look at them as we want them to be styled, which is a sphere. Now, that may have been a lot right there, but if we break this down into those 3 segments, we first need to look at the chromophore. So all we have to do is add a ball and stick representation. But if we do this, then that means that every single atom will be represented this way. And here's where the nodes become so powerful, as we can simply select for a single residue ID. And if we do that, and if we know that the chromophore is residue number 66, we can then just select for this one, and then connect the selection to the selection on the ball and stick. And there you go. So that is just the chromophore being styled right there. And it is in gray because we are currently in solid view, whereas Blender offers these multiple views based on your computer's power. This rendered view is usually only beneficial for just looking at the final product. Now to make it glow, for instance, we can add a new material on the right here and add an emission to it. And if we do this, and if I just rename it chromophore just for simplicity's sake, we can change the material of the style in the workflow to chromophore, and it can glow. And if I increase the emission strength to control how much light the chromophore emits, we can see this process take place in real time. So this is useful for emphasizing key residues in these different models, such as a specific amino acid. Now, Next, for the cartoon representation, we can simply add this within the same workflow as the ball and stick. So if we add a cartoon and connect the input to it, however, there is only that one output that we discussed earlier, but there's an easy workaround of this. If you just hit Shift A, and then you start typing join geometry, and Shift A is just a shortcut for add. So this combines both the cartoon and ball and stick styles into the single outputs, allowing us to see both at once. Now the scene is very dark, and this is because we have no lighting in the scene yet. So if I just simply added a sun, and we can change the angle of its direction of light towards the protein. So we can play around with that and have some fun. And we can also add a set color node. So we can change the color of our style in the workspace instead of separately like we did with the chromophore. So I can drop this in, and this will only affect the color of the cartoon style as it precedes it. And we can play around and differentiate different regions of the protein. So now we have the chromophore in the backbone. For the electron density, I have sort of pre-added in some nodes here to save some time. So I first add a style surface node, which serves as the base for our electron density visualization. We can then convert this into a volumetric representation using mesh to volume. And lastly, a style density wire node. It gives us a little bit more control over it. So if I just simply plug these in, I also added in a pre-material, just so I didn't have to go through that process again. That is titled density, and this is just a blue emission color. So if we adjust these parameters, we can then fine tune the density levels, making the visualization appear how we want to. Here, I'm just going to adjust the radius to demonstrate that. So that may have been a lot just thrown at you, and I will kind of go over a little more specifics in a second. But to just sort of take another look at this, we first have these three different paths that start off from the input. We have the ball and stick, which is the chromophore, and we have residue 66 selected. We have the cartoon style, styled as the color red there, and then the electron density styled as the surface. And this modular approach shows how flexible this geometry nodes can be for visualization, allowing us to sort of mix and match and customize the layers depending on which we want to emphasize. So that can be very convenient. And you can also have a lot of fun with this. As for instance, I did a one-to-one model of a Coca-Cola advertisement that nobody asked for using this exact PDB file and molecular nodes. So please enjoy it for the next 6 seconds. And thank you very much for watching that. That was quite fun to make, actually. And that didn't even take that long. Now to go over what we just discussed, as that may have been a bit overwhelming, let's take a look at just the styling options that are available to you in Blender. So you have pretty much 5 general ones that you will ever use for looking at a protein. You have this sphere style, a ball and stick model, which is just the spheres showing the bonds between one another. You can also have a cartoon representation, after I zoom in here. A cartoon, or a ribbon style, and finally a surface style. The surface style is quite computationally heavy, so I would just beware of that, as it can sometimes crash Blender. But you could also decrease the quality or play around with the parameters of it if you'd like. So those are the styles, but in order to play around with what you want to see specifically, you have to use these selection nodes. So if you look on the left, there's a bunch of different options you can choose from, such as selecting by chain, element, residue name, residue range, whether it's a lipid, a solvent, a backbone, side chain, etc. So to go to the very basics, if we look at a very simple protein, such as keratin, using this PDB file 6EC0, we can use selections to refine this visualization by selecting specific characteristics. For instance, on the workflow over here on the right, I have this select chain node in red, and this can allow us to isolate individual protein chains from this data set. So if we connect this cartoon representation to it, we can selectively apply this style to specific parts of the molecule. And you can notice how it sort of updates automatically based on which chain we are electing to see. And next, if we wanted to look at the residues of each of these chains, in addition to just the backbone cartoon style there, we can simply add a ball and stick representation to the top of it, where we only display atoms from chain A, and chain A is visualized in the pinkish red there, but we do have chains A and B shown for the cartoon. So that's a way to selectively style different proteins. Another fun thing you can do with this is if we were interested in which atoms or residues were within a specific distance or region from one another. So one method you could do this, which is shown at the top, is you can add a select sphere node to the ball and stick representation. And this allows us to define a specific region in 3D space and focus on the atoms within that area. And we can scale and move this sphere to dynamically control which atoms are included. And this is very useful for highlighting key binding sites or active regions within a protein. Now we can also combine these selection nodes, which is very powerful. So instead of selecting atoms based on spatial position, we can refine this image by isolating a specific range of residue IDs within chain A only. So I simply just got rid of that select sphere node and added a residue ID range, and it is connected to the ball and stick style node. So we have select only chain A and show only residues 254 to 269. And we can dynamically play and define the start and end points of this range in real time and focus on a particular section of a chain. So that's also very useful. So that was a quick example of this, looking at this compoundment of these nodes. And this can multiply and you can add as much as you want. So if somebody told you to show me chain A specific amino acids between a residue range, we can do that ourselves actually. So we have a chain A coupled to the same residue ID range. However, we can also select for certain residues within that range. So you can see here, I'm just kind of clicking around and just showing this process in real time. So here's sort of a real world example where somebody told you to show me the nitrogen and oxygen atoms of only arginine and aspartic acid side chains between residues 239 to 260 on chain A. I do not know why anybody would ever ask you to do that, but this is just a way you can do that. So on the far left in the workflow, we are only selecting for nitrogen and oxygen and residues arginine and aspartic acid that are side chain atoms within that ID range, followed by chain A. And this is all connected to that ball and stick node or styling node at the end on the far right. And then if on the left, you can see these nitrogen and oxygen atoms shown in the blue and red there. So that's just kind of an interesting look at this compoundment of nodes that you can do. And you can also go quite crazy with this. As I kind of show right here, as this sort of animation I made on the left was done using this absolutely insane looking workflow that I do with no organization at all. So that is partly on me. But this is just a cool look at sort of the complexity that you can derive from this. So those are the style options that you can do. The coloring options are, they work essentially the same as selection nodes. However, you do use a different sort of Boolean for it or connector. So the set color node here just comes before whichever style that you want it to look as, and you can directly play around with it. Now, if we connect a color node to that set color node, which is in yellow instead of pink, we can, immediately and in real time play around with the style that we want. So here I just connected a rainbow color selection to the color node. And then some more things you can do. So if you wanted to visualize specific atoms, but wanted to color them by their actual identity, say if we added a ball stick representation to this set color node, but this means all of the atoms will actually be colored the exact same as that chain it corresponds to. But if we don't want that, there is another option you can do where you can add a color common elements node. And by doing this, you can actually only color, so you can selectively choose which atoms you want colored which way. And if you connect that original set color by chain node to just the carbon input of this common elements node, you can actually just play around with the color of the carbon atoms only based on which chain they are on. I just confused myself a little bit there, but that's essentially how that works. And you can kind of see me play around with this process. So because the carbon is connected to the color chain, when I play around with that, as shown here, you can see that, yes, all the carbon atoms get changed. However, because nothing's connected to the oxygen and nitrogen, if I were to change those get changed universally. So I hope that made a little bit more sense. Now for DNA modeling, this is also a very useful tool. And this comes from the use of curves where you can custom generate your own DNA molecules. So curves are powerful tools that allow us to create these smooth shapes. And this will act as the backbone for a DNA structure, as they are very flexible and can be easily adjusted. So if we use this curve as a guiding path and create a new node system for it, as shown on the right, we can project these established structures of DNA onto this as a ball and stick representation. So here I am just adding in the required nodes for it, as molecular nodes has its own sort of segment for DNA, which is very useful. So yes, we just plug in the DNA double helix, just drop it on the line, and it'll automatically input it, but we need to connect this bases input as well to it. So you can see right there, we have our own DNA, but it's only styled as vertices right now because we do not have any, we do not have the ball and stick styling connected to it. So once we do that, boom, there we go. And you can play around with the coloring. And the most useful thing about this is if you edit the curve, it will update in real time as well. So you can play around with the settings. And yes, if we just make our little viewport bigger, you can go to the edit mode, play around with the location of it, and the DNA updates automatically for you. You can extrude more vertices on it, rotate it, scale it, however you want. And the DNA double helix node is very beneficial for animations of DNA, as it has many different settings you can play around with. So I can increase the length, I can extend the helix, I can create a longer DNA fragment. I can also adjust base pair spacing to refine how nucleotides are distributed along this curve. So that's a very fun, useful tip and trick you would have. And lastly, the animations, which are very useful in Blender, are actually surprisingly easy to work with. So if we go back to our keratin model, we can first shrink this node section just so we're not staring at this the whole time. And then if we add a different section, such as a timeline shown at the bottom, if we hit the space bar, that just plays the animation. And then what you can do is if you right click it and hit insert keyframe on any object, this isn't only limited to molecular nodes, and then just increase the timeline, move it around a little bit. And if we hit the shortcut, which is just I, that will insert a keyframe, hit play, and it's that simple. And this can work in conjunction with scaling or even a rotation. So if we rotate this as well, go back to the start. And if we hit play, look at that, quite simple. And then if you do this combined with one another, so if you move it and rotate it, it will do the same as well. So just one final time. So that's kind of a cool thing you can do. You essentially just have an object at a certain frame within the animation, insert a keyframe, move the timeline, and then move it to wherever you want it to be. So that was kind of a brief overview of the things you can do in Blender. That is working with the actual molecular data. However, you can custom make your own lipid bilayers, for instance. One interesting thing you can do with Blender is to color the residues based on their actual charge or properties. So the hydrophobic residues on this picture on the left are in yellow. We have the polar in blue, positively charged in pink, and negative in red. So this is an interesting look at an amphipathic helix that can be wedged into a lipid bilayer, showing the hydrophobic and hydrophilic residues that is not possible in Pymol. And on the right, I have a type 3 secretion system, which is a bacterial membrane protein that is used to transport proteins across 2 different membranes. So this just demonstrates how these molecular machines can integrate into membrane systems, and you can visualize this process as well. You can also generate some very artistic illustrations for communicating different concepts, as science is not all about raw data, it's also about storytelling. So by using Blender's rendering capabilities with this artistic design, we can make these compelling visuals that explain complex concepts in an intuitive way. So the stylized depiction emphasizes these structural differences between rod and cone cells on the left, and on the right, we see an illustration of CRISPR-Cas9, which is essentially scanning DNA to find a sequence that it matches in order to bind with it. And this was done using PDB 6VPC. So these types of illustrations are useful not just for research, but also for public outreach, education, and journal covers, which helps bridge that gap between complex science and broader audiences. And by using these artistic tools alongside scientific accuracy, these powerful visuals can make molecular and cellular biology more accessible. And I made this fun little thank you animation at the end here. So I just wanted to thank you all for listening to me go on about Blender, and it is something I'm very passionate about, and I'd love to talk about it. So thank you very much for listening. We have some acknowledgements that we would like to share as well. Helena, if you can start.
Yeah, I'd just like to really thank my lab for giving me the opportunity to learn all of this, for my supervisor, Dr. Stranadka, and everyone who has been kind of foundational in my education as a scientist and in going through so many iterations of all my figures and learning how to use these programs.
Yep, and I just wanted to thank my lab as well, as they are a very supportive group. I'm very fortunate to be able to be in this position. And we also wanted to extend our thank you to the IUBMB for hosting us and helping us so much getting this all planned. And with that, we also just have a shameless plug here at the end, which is, yes, they mentioned we founded this science illustrations sort of service called SHS, and we have our website shown there, which is links and our relevant names as well. So that all said, thank you very much for listening, and we'll open the floor to any questions again.
Thank you so much, Stephen. So now we have one question in the chat already. So what kind of computer specs are needed to do all of this in Blender?
Yeah, so the minimum specs are 8 GB of RAM for your computer and 2 GB of GPU. I'm not totally sure what GPU really entails. What I can say though is that I've done all of those animations on a MacBook Air with only 8 GB of RAM. So that is kind of what I'm limited to. So you can make virtually anything that you dream of. But yes, I'm able to do those on just 8 GB of RAM. So one day I want to upgrade, that's for sure. But yes, so you are not limited if you have somewhat of a not very up-to-date computer. I originally started to learn on a 2014 MacBook Air, and it still worked there as well.
All right, perfect. Thank you. Anybody else have any questions? You can type it in the Q&A chat, or you can raise your hand. I do have a question, actually myself. I work on like mechanical proteins, so like opening and closing of proteins. Is that something you can actually model like on Blender, like the actual like opening and
Yeah, so
closing?
you can either upload molecular dynamics data in that sort of window panel when you originally upload the PDB file. You just essentially hit the drop-down menu and change from PDB to molecular dynamics. With that, though, you do have to have the data yourself. It won't take it from the internet for you. And then if you weren't doing molecular dynamics, you can also import two different, say an open and close conformation, and you can blend those two together in real time. However, I don't believe I probably wouldn't use that for like a scientific publishment or anything like that, but just for visualization purposes for that would work.
Yeah, just for like the visual, like
Yeah.
to think like a movie almost of what I'm talking about from the.
Yeah, it is possible to morph the two states together.
And also to that point, you're also able to do some basic visualizations in Chimera X. You can create movies there as well. The things that it's not nearly as good as it's not nearly as good is if you need to do like a lot of moving around, like Steve was showing, like with the a beautiful example of the secretion system. Like you can't like unwind a protein, or you can't make proteins glow. But you can like shift a protein from one conformation to another, and it'll like link those up and create a video of that in Chimera X as well.
Okay, yeah, that's cool. Okay, some more. Someone has a comment that Blender scares me because it has 1,000 buttons but looks so powerful. Thank you.
I mean, actually, I do have a comment on that. It does look very terrifying. However, I am not much of a coder or a scripter. So you can kind of think of it as the same thing or very similar to Chimera, but instead it just kind of forgoes the need for any coding. It just uses those sort of nodes that you select. So it's a bit more of a visual way to sort of design this model from scratch, let's say.
And someone asked, like the Coca-Cola ad you made, can I use Blender for other visuals, not
Yes.
molecules?
I have made a lot of random images and photos that are not even related to science at all. You can make some pretty funny things too. I made a bachelor party image of a bunch of different things, and they look very cartoony and interesting. And there are a lot of add-ons as well that you can download that import or have pre-made objects for you, such as like a glass of wine, a bottle of water, an iPhone, that you can just upload directly. So you wouldn't even have to make them from scratch.
That's actually really cool. And Spencer asked, what would you say to someone afraid that learning to make Blender figures during graduate school would take too much of their time?
I mean, I guess I might have a lot of free time on my hands because the learning curve that I had was, it was a little difficult. However, I feel like I just wasn't watching a lot of tutorials, as I just told myself, I'll just learn by trial and error. But the process is quite simple. Like that Coca-Cola ad, for instance, I think I made that in 10 minutes. The render itself took like 2 days of just like loading all those frames. But to put all those nodes together, if you know what you're doing, essentially, it's honestly very fast. But yes, learning curve is quite scary. It looks terrifying, but it's really not. It's just connecting these different things together.
And I would also say that, I mean, graduate school is like a really good time to like take a little bit of time to go through and learn, you know, a hard skill that is going to be useful, maybe in other parts of your life or in kind of translatable to other things. So I would say if you give yourself like a small task, this is how I learned Illustrator, which also has a bit of a learning curve, not as bad as Blender's. But like give yourself like, try to make one thing, like a specific thing you have in mind, and you'll go through, and yeah, it'll be a bit time consuming with tutorials, but the next time you do it, it'll be like 10 times faster. So first time sucks, but the next time's way easier.
I will say also, this is addicting if you get into it. I've spent a lot of nights just up until the wee hours of the morning, just playing around with it. So be forewarned.
And another question is, do you have any recommended resources or videos on the basics of how to use Blender?
Yes, the Brady Johnston, if you just look up his name on YouTube, or even if you looked up molecular nodes, but Brady Johnston in particular, because he is the creator of it, he has a lot of brilliant introductory tutorials on it. And he makes it very clear. He goes very in depth at some points as well. But he knows what he's talking about, or is very good at introducing the topic. Sorry, I should say. Brady Johnston, that's his name.
And I know you just were talking about that there's add-ons. Do you get those from like the website, or those things that people are posting on like GitHub repositories, etc.?
Oh, right. I guess I should have clarified that. Yes, there are some where you can Google, like if you just Google Blender add-ons. However, in Blender, at the top beside the file button, like if you want to hit file save as, if you just hit edit and then preferences, there's actually they have the official add-ons are already sort of preloaded in there, or you're able to just import them from the internet directly by just hitting like a little checkbox beside them. So for this molecular nodes, for instance, You don't have to download anything directly from the internet. If you just go to edit in the top left, once you have Blender open, and then if you have preferences, there'll be a side panel saying get add-ons, hit that, and molecular nodes should be an option there that you just hit the little checkbox for on the left and you're good to go.
Okay, perfect. Thank you. And is there any last questions? This is the time. And you can also raise your hand if you want to speak instead of typing.
Oh, and we did just, to the point that was said, want to say that basically every program we talked about was free other than Illustrator and Prism. But Inkscape's a really good alternative to look into. So, yeah, they're all free, they're all open source. And Blender, Chimera X, Primal, Coot, lots of tutorials and lots of resources for your work.
All right, thank you so much. Thank you so much. Let's give a virtual round of applause to Helena and Stephen. And we're going to turn it back over.
Okay, well, that was a lot to take in, but it was extremely helpful. I know for us graduate students and researchers of all levels. So to everyone, that concludes our PABMB event for this year. So a huge shout out again and thank you to our speakers for their efforts and time to make this an eventful webinar and workshop. And I'm sure that these tools that you've spoken about are now kind of waiting for a bunch of people to sign up and use them. So thank you also to all of you for showing up and engaging with us in our event. And thank you also to the IUBMB and the IUBMB Trainee Initiative for all of their support. We also have a number of previous events posted on the IUBMB website under the past events tab, if you search up the IUBMB Trainee Initiative, and are interested in what we've done as well. And finally, because we are always on the hunt for funding opportunities, we have listed a few over here as supported by the IUBMB. So Elisa, if you could go to the next slide, please. Thank you. So the ones with the most immediate deadlines coming up are 2 travel fellowships, the ProLab Scholarship for Latin American scientists wishing to do an exchange term in Europe, and also the Wood Whelan Fellowships. We also have more fellowships listed on the IUBMB website, so be sure to pay it a visit for more details. And you can also sign up for our monthly newsletter, which includes all of this information. And yeah, thank you again and have a great weekend wherever you are coming up. And thanks again for tuning in.